if I told you that if someone were to cut off your head, it would regenerate within just 7 to 10 days? Enter Planaria, a fascinating creature known for their regenerative capabilities. My name is Ellen Mayer, and my research aims to investigate the effects of pseudoephedrine, a primary ingredient in the legal manufacture of methamphetamine, on the regeneration of Degisia directocephala planaria. Using morphology and behavior as endpoints, researchers at Swarthmore College and Westchester University have established a negative correlation between high concentrations of lethal narcotics and planaria. Although current research has investigated the effects of drug overdose, pseudoephedrine has yet to be tested, therefore substantiating the need for further safety evaluation. Ultimately, my goal is to increase our understanding of the potential side effects of nasal decongestants and its impact on wound healing by posing the question, to what extent do varying doses of pseudoephedrine affect planarian regeneration? As the difference between these two images. The first one, when you walk in the pharmacy, there are medications naturally lining the aisles. These medications are referred to as over-the-counter medications, which are easily accessible to the public. However, if you look at the second image, there are medications that are kept behind the counter, which are less accessible to the public. And so, working in the pharmacy made me question, what is it about these medications that permits more restrictions compared to typical over-the-counter medications, like your ibuprofen or your Tylenol? And so through further research, I discovered that there's an active ingredient in this medication which permits its restrictions, leading me to ask, are these medications inherently addictive? Does the nature of these medications cause any harm to the human body? Hi, my name is Ellen Mayer, and today I'm telling you about my AP research project on cold medicine, worms, and wound healing. By first asking the question, to what extent does pseudoephedrine hydrogen chloride, or Sudafed, affect the regeneration of Degisia tegrina planaria? And basically, and simply put, pseudoephedrine is a type of cold medicine, and planaria is a type of regenerative worm. It's okay if you don't understand too much of the terms right now. I'll get more into what everything means in just a second. So throughout my research project, I'll be working with Dr. Annie Cunningham from the University of Southern California in order to investigate this question. And for a quick overview of my experiment, I'll be first amputating the planaria, then expose them to different concentrations of PSC, and then observing how this concentrations of PSC affect the regeneration of planaria as it starts to recuperate its cells. Now, it's quite fascinating actually. I'll talk a little bit about the nature of brown planaria itself and why I chose to study it as my model organism. So planaria is a flat worm which originates and grows in freshwater ponds or also naturally as an aquarium parasite. It's really cool. Planaria is composed of something what we call 20% neoblasts. And neoblasts are essentially just stem cells. So if you were to cut the planaria at certain parts, stem cells would aggregate in that area and go through a process called differentiation, where it would specialize and form something called a blastema in order to rejuvenate the damaged and lost cells in this process. And it's very fascinating because if you were to cut a planaria in even 279 different parts, every single one of those 279 parts would regenerate into a full-fledged worm. It would be able to regenerate its eyes, its brain, its head, and all. And so let me talk a little bit about my pertinent surrounding literature regarding my topic, starting with the first study I investigated. So Le Carré and his colleagues deduced the dangers of pseudoephedrine and its nature. So they were able to conclude that although PSC is an effective vasoconstrictor, and essentially a vasoconstrictor is basically a medication that constricts your blood vessels, therefore allowing for easier ability to breathe, which is what cold medications do in order to help us when we're sick. But however, he also found that pseudoephedrine in itself has severe adverse cardiovascular and neurological side effects, which through his studies that he investigated, it could produce side effects such as stroke or psychosis or even hypertension and possibly even death. And through the study, I deduced two different things. The first one, I was able to verify the dangers of PSC itself. And two, I was also able to devise my hypothesis in order to investigate why I think that PSC as a natural stimulant can potentially increase the rate of regeneration. Moving on to the next study, which was done by Levin and his colleagues, he studied a medication called ivermectin. Ivermectin is a type of medication that people use when they have head lice or body lice. And it's supposed to fight the parasites and kill the parasites on your body. So Levin and his colleagues found that a greater exposure to ivermectin essentially increases mortality rates in the planaria, as seen by this graph. So the planaria constantly died under this exposure of this medication, and this allowed me to kind of conclude 3 different things. The first one was to verify the use of planaria in my study as an effective model for regeneration. 2, also devise my methodology, since this experiment is very similar to my own, except that it uses a different medication in order to study the effect on planaria. And the third part is also to study the different stages that planaria takes when it does regenerate. And so this established my gap in the research. 1, planaria is an effective model, but also 2, PSC is able to, has a lot of different dangers that needs to be investigated, since it's also a precursor for crystal meth or methamphetamine. And also planaria, since it's never been tested on PSC, that is exactly the gap that I'll be studying in my research. Okay, let me get a little bit more into the methodology and how I decided to conduct my experiment. Starting with the first step. So I took a general approach to this experiment by taking upon a mixed qualitative and quantitative correlational experiment. Essentially what this means is that I'll be analyzing qualitative variables of each of the planaria throughout its regenerative process, such as observing the behavior of the planaria, the shape, the color, and how it looks and changes. Also, I'll be observing quantitative variables, like the rate of regeneration, and also the length and the width of the planaria. It'll be a 25-day study where I'll be amputating the heads on day 5. And so for this first step of my experiment, of course, is to obtain the materials. So throughout the study, I also bolded each of the materials that I listed on the picture over here, but I'll be using and utilizing 2 different microscopes for this experiment. The first one being a digital microscope. This will be for the general observations of the planaria, how it looks and such. But for the compound microscope, this will be for more accurate and precise measurements in order to make sure that I cut the planaria at specific lengths. In order to ensure that my measurements are accurate, I will also be using a micropipette in order to dispense the correct milliliters, which is 8 milliliters of each of the Petri dishes. And lastly, of course, I need to obtain the planarium itself, which I'll be obtaining from Carolina Biological Supply. Okay, next step of my procedure is to split up each of the Petri dishes in its respective groups. I'll be having a total of 5 different groups, 3 of them being experimental and 2 of them being on my control groups. I also put down my predictions and my hypothesis of each of the different concentrations. So with my experimental groups, I'll be testing a low to high concentration of PSC. And then I'll also be having my negative and my positive controls as listed on the screen. Because PSC is a natural stimulant and it's supposed to produce a sense of euphoria, usually in human beings, I hypothesize that planaria, the rate of regeneration will increase to a certain point, but at a certain point at high concentrations and over chronic exposure, I hypothesize and I predict that the rate of regeneration will essentially decrease, leading to greater mortality rates in the planaria. Next up is decapitation. And decapitation for the planaria will compose of 2 different sub-steps. The first one being immobilization. To immobilize the planaria and make it easier to cut it at a more precise angle, I will be using a method where you place the planaria under ice in order to slow its movement down. The next step will be to create a horizontal bisection to amputate the head and the tail of the planaria, right under where the eyes are located. And the last step of my process is what I call the regeneration assay. So in order to study the regeneration, of course, I'm not only going to be looking at how long it takes to grow, but I'll specifically be observing 3 different factors and how they specifically regenerate. First one being the digestive system. If you place the planaria on a white surface, because of its translucent nature, it is very, very easy to deduce the dark colored digestive tract, which I'll be observing. For the pharynx, if you place the planaria on a dark surface and with the addition of food, you can watch as the planaria extends its pharynx out to grab the food and bring it back in. And then the last part, of course, is to observe the regeneration of its eyes. Since I'll be cutting off its head, I'll be watching as the planaria increases its regeneration rate of the rest of its organs, its eyeballs, its brain, and so forth. So if you place the planaria on a dark surface, if it does have the presence of eyes, that means it'll move towards the dark, which I'll be able to observe. And so my experiment continues. Thank you.